helping to do a fast track of how to develop such strategy, because we learned a few things, and I hope I could all put that in the slides. Yeah, so the first part, the first word of my title, the second part of, like of the subtitle of my talk is community. And of course, this is a community effort. I'm actually not the main head behind it. It's the main person behind it that spent most of his last 3 years working on it is James. James, together with Ida and Maxime to some extent, and also Christina being generous enough to sponsor both James, Maxime, and I think are the main people behind it. But since James and Ida have had recently another baby, Theo, in August, I'm kind of taking over as the representative of this project while James is on paternity leave, and so I feel myself kind of the chief whip while in the absence of the party leader here to kind of make everyone run. However, next to these 4 people I just mentioned, there are many more. So here you see actually an overview of all the 71 individuals that signed up at some point to the SPAM community during its existence. And signing up means there's also maybe more loose connection about it, but overall, these are the people who have a GitHub account and were at some point planning to contribute to some way or another to the SPAM community. And overall, what does it mean? So 70%, 49 out of 71 SPAM members that you saw on the slide before managed to contribute code to Ancient Metagenome Deer. In total, we have almost 10,000 commits. I will talk about this in a second, what this means. And the project started with its first contribution on the 26th of July, 2020, and is still running. And the last contribution has been made last week or a week and a half ago in beginning of December. Now the question is, how did we actually come about this? And James had written an anecdote about it a bit in the SPAM blog, that SPAM community blog, if you are interested in all the details. But more or less it started when this later manuscript, the idea for this manuscript was conceived by Ludo Galando, reaching out to other PIs at the in the ISPA community, Ancient DNA World, because Nature Review asked for a methods primer about ancient DNA analysis. And at the time, as you see here, the last author of the paper is our boss, Christina Warner. And then she came to us, to James and me. We want to show typical analysis of ancient DNA. And for my part, I contributed here on the bottom, the figure panel 4C, by doing some metagenomic microbes analysis from some samples. However, there was a second question that was asked that was much bigger than this one, and there was like, how many ancient metagenomic samples are there out there published so far? So at the time it was 2020. Where were they sampled? So where were the location where they were collected? So are they from Europe? Are they from the Americas and so on? And how old are these samples? And the big caveat was more or less, we would have liked the answer already yesterday. And so James was actually asked by Tina, James was at the time a PhD student with her, whether he knows all of that. And then James actually realized that it's not feasible for a one-person job to do that. So he went ahead and came up with a strategy to include the community. And the outcome is more or less this map that you can see here. You see these kind of rectangles, a bit hard to read on the slide, that are showing the locations of samples across Europe for humans, animals, plants, microorganisms, and pathogens separately. And the end of it, while this paper was published in January 2021, by now we're still going 3 years later, and we have recorded almost 2,000 samples from 85 studies and referring to 6,075 sequencing data files. So I could stop here, But I thought I will take you instead on a journey and walk everyone through a bit slower. And the first journey, and trying to also explain while going through the journey, the different concepts of this collaborative project, what were important things that we took from it, what we think is important, and things like this. So on the first chapter here on our time axis timescale is, like I said, the first day, 26th of July 2020, when a GitHub repository is created. What does it mean? So for people who are not familiar with GitHub, usually GitHub allows uses underneath the command line tool Git, but provides a very clickable, searchable visual help to actually view the content of this version control. So Git is a version control program. And usually you have here on the left-hand side, you see that all the files that you want the version control system to track, you have here. And then usually the most important file of the, at least for newcomers to it, is the README, which then is here shown on the right-hand side that explains all the content. So this is ancient metagenome deer. What is the idea behind it? What kind of samples to recover? It has a description, a usage citation, and all of that for people to come. You also have some statistics, who has worked with, like who are the contributors, how much code was provided, and so on. But overall, if you take one step back and say, okay, Alex, not so much details, too many details, if you kind of do it in Star Trek terms, it's the bridge. So GitHub is kind of helping us organize the whole project by itself. It allows us to communicate with people, let them allow to add their samples, their data to this repository, allows us to review it. If we realize someone did a mistake, we can correct that. All of this is done within GitHub locally. As well, like locally, we also use Git for that, but actually the whole website is there. In theory, everyone who is interested in collaborating with us can go there and contribute to the one. So that is one thing that we said very early on. We want to have a resource that is accessible around the world for everyone. There's no restriction that it only runs on a certain institute server or something like this. And overall, we realized that the most popular such resource is GitHub. And with every, like, the first thing always is like a contribution to GitHub is a commit. So if you see the term commit here, that's a contribution to GitHub. So you say, please track my file, and if I add new stuff to it, please be aware of it that I want to update something. And you'll see traditional initial commits, the big start of the journey, like push the start engine, like the start the engine button, where James said, oh, here is our repository containing lists of all the published ancient metagenome samples and related, which over the years grew to all of this kind of message that you saw here on the right-hand side. What happened in the first few weeks there was that, like I said, it's a version control system, so people can add and data to their to the repository. In the beginning, very much because we were at the time based in Jena at the MPI for Science of the Human History. Mostly it was people within the group, so James, Maxime, Irina, who contributed. And what we also saw is like it started at the first day with in total 5 contributions, 5 commits. On the second day, it was 2, but then slowly but surely, we kind of gained up speed and many people contributed things. And from the name of the things, it was also a lot of foundational work that I will talk about in a bit later in the journey. The first thing that has been added in for the journey was that all the samples, so more or less what James did is that he asked people to do a literature review of the papers they know and categorize all the samples that are described in these manuscripts in 3 categories. We have environmental ancient metagenome samples. So if you have sediments, for example, ice cores or something like this is one group. Then we have host-associated ancient metagenome samples. That's, for example, dental calculus, with Sandra and James work mostly on, Irina as well, or paleofeces, what is my home turf. And then finally, we also have host-associated ancient single genomes, which is usually isolated genomes of pathogen work. And we then generated a list of minimum information that we think was important to collect. So there were some general information, project name, publication year, publication DOI, so that we have also linked to original publication. What's the name of the site, longitude, latitude, geographical location name, and so on, sample name. So most of these things were mainly driven by the question that Tina wanted to know. What's like the study? So how many studies do we have? Where are they based? Latitude, longitude, geographic name, and how old are they? Sample age. But however, we had also for the individual categories, we also then later decided to add a few specific terms like primary study focus. So for example, is it a sediment study? Is it a plant-based study for the environmental samples? Sequence name, or the depth of the sampling, if you, of course. What is the sample host? Did you sample human? Did you sample a chimp or gorilla? And is it oral? Is it a stool microbiome sample, or what is the pathogen domain in the end? So we have added a lot of a list of information, which was then for every study the people went through it and they and added these things to it. And because we realized very early on, so already 5 days later after the first initial commit, that we don't really will manage the four of us actually to really add all these papers, because we're also not so familiar with environmental samples or pathogen samples, that we started to recruit people in a sort of and brief them in a metadata hackathon. So what James did is, at the same time, James and a few others of us at the time were organizing a workshop to take place at the ISPA in Toulouse. That was then later postponed, so we did it online. And so James had a list of individuals who were interested in taking part in this online workshop. And so he then sent out an email to the 63 people that later attended from 15 different countries. And in the end, 17 of these individuals said that they want to help with this project and contribute. So we made a list of publications and things like this and went through it one by one. This is a sample. What are the information? Can we track them and add them to the part? However, as I said, this is a version, like GitHub is our bridge, but under the hood, it uses a command line tool, which is called Git, and therefore also named GitHub as a web interface to Git. And for most people, when you are more on the techie side, on the computer science side of things, you might do your code revisions or something with Git. I can very much recommend it to do version controlling with Git if you're working on code, R code, or writing analysis scripts or something. But usually it looks like something like this here on the left-hand side. And the problem, while Git is very cool, it allows you version control. It allows you to that multiple individuals work together at the same time. It doesn't matter if someone tries to add study A to a table and another person at the same time tries to add study B. All of this is taken care of, not as nice as maybe Google Sheets would do it, but it works. It's still kind of, it's a bit of a demotivation for people who are not tech savvy when they see this kind of complicated command line interface, which is not always straightforward. So how did we actually get around, how did we lower the barrier that people actually contribute who have never worked with Git and are not familiar with all the tools and with all the hacks of GitHub? The first thing that we did is, and that's mainly James and to some extent here Megan is also helped him revise the material, is to really write the extensive teaching material for GitHub. So he really wrote it all down, did actually training, one-on-one trainings, teaching people how to use it, how do you do certain operations, how do I add my new data, what do I do if someone added something in the meantime, how do I merge the things and so on things. So James did it, and we publish this whole teaching material as part of our spam community that we have, and the Introduction to Ancient Metagenomics book, if you want to take a look yourself, which was very helpful. The second part is that we have also in the background a Slack community running. So the spam Slack community, where we have a dedicated channel. And for a lot of these things, we then also discuss issues that occurred, how you deal with this. Oh, there's something unclear, they're ambiguous. The authors didn't really write things very clearly in your material. We then have an open discussion and discuss this around it. Oh, and James giving here advice. The last thing is we also showed that next to this command line interface, for a lot of the basic stuff, the functionality is actually, you don't have to really use the command line interface, but there's tools like GitHub Desktop, for example, which allows you a click and drag and drop interface to GitHub to actually do the same operations. Just you don't see the code, so to speak. And for many people who were still a bit fearful of like sitting there in front of the terminal and typing in the commands, that really helped as well, because it has this visual interface. It's kind of showing in a condensed way what you would later also see on GitHub itself. And so we could help them. And by using the strategies like teaching material, being there for direct help, and having alternatives next to the command line, really enabled a lot of people to actually help us to add to the material. And so how did we actually add data? That was, like I said, we did a literature review for every news publication. We do an issue, because GitHub has a few additional features that Git doesn't have. One of this is an issue tracking. So usually they're used to, oh, you wrote code. Yeah, try to run it. It doesn't work. Please fix it. But we used it in a way that we, like you see here, have a few publications, Guns 2013, Paris 2013, Corona 2013, and a few other things where we kind of say, oh, this is a new publication here by this yellow badge, easily identifiable, where we track what we have added and want to edit. So if it's open, so you see the open ones, then means we haven't worked on it yet. All the ones that have been added are then later closed and are here among this 444 ones. It's always important to have one issue per study, so you can really track each issue over time. And we also have here on this column here on the right-hand side, you see that individuals assign themselves to kind of claiming responsibility, and it's also tracked over the future, which person has been responsible for adding the study to it, so you don't duplicate work or someone takes false claim credit for other people's work. And when we find a new publication, then usually we create an issue, which looks like this. So we have a template that says, here's the link to the study. Maybe if they're in the later part, also where's the sequencing data stored, I will talk about in the last bit. Adding some information, what kind of group it is from, do we have some additional information to also make it the users easier to kind of identify them, so that people from the pathogen field might be feel more obliged to actually add pathogen papers because they benefit from them more than someone working on plants and so on. And so we have an easy tracking system. Once you edit all the data and you think, okay, I went through the whole literature and did everything, then we do something which is a request for merging the data. So taking the old table and kind of appending your data at the bottom of it, which in GitHub speak is called pull request, where we again have a template that has some information which you have to check out, and that's more or less how it looks like. So you more or less ask, please, people, I want to add data, and the important part here is that you can then also see once you do it, for everyone, what did you want to add? So in this case, all the pluses here in green show, oh, here are the 5 samples of the study Parker 2023 that want to be added by the user. And at the top, we see all the files that have been changed. So we change usually the samples nowadays in the library table, but we also change the change log to keep track of what we're doing. So this is usually what you see as a person when you check out this pull request. And then the very important thing is that you can't, or the idea is that you don't add something automatically. However, we have at least the 4I principle. So another mostly senior individual, James, Ida, I, a few other people, are then going back and double checking what you added by reading through the publication ourselves and making sure that we agree what you came up with. And in this case, for example, James said, okay, here, please do that and comment, but overall everything is fine and we can do it. But without his approval, it's impossible for the person to actually merge it. And that's also important that you don't really, because of this version control and this kind of 4I principle, you reduce the risk of losing data, first of all, plus also adding wrong data that you might drag on forever because someone thought it's funny to change something in the database and with then something slipping it. Additionally to that, at some point we started also to implement automatic checks. So what are these automatic checks? So for example, we have a tool that goes through the different columns and then says, okay, the longitude and latitude values, they have to be numerical. You can't or an A if they're missing, but they can't be a site location name. So for example, you forgot to add a column by accident in your table and then everything moves to the side. You have suddenly a shift in the columns that would be automatically detected. We also have the thing that we check if the country name is valid. So for example, one of the examples we had, Scotland is not a valid country name according to the thing. It's the United Kingdom instead. So if all these checks run through, that is like for checking the dummy thing, everything fine, it can be first approved, and then finally it's merged, which means now for the next person, the final version, the current version of the table has this additional data, and we can continue. And finally, what we have been realizing is that how to get more people working is also to have dedicated meetings. We call them hackathons or metadata-tons, where I really dedicate hours to be very productive. Sometimes we do it with local groups. So we say, oh, 3 people from the same institute want to sit together and work for 5 hours or 4 hours on some questions to add data there. Then we do it, but we also had long meetups on Zoom and GatherTown over time. I stole one of these screenshots from GatherTown because James is an extreme big GatherTown fan, which was not from one of the hackathons, but the summer school we did. But it kind of illustrates the thing that there's many people you have that interact together. You have breakout rooms, you can chat about problems, and you really get things done. So the end of this whole story after adding all this data was that at 5.5 weeks later, on the 3rd of September, we were able to actually release a preprint, and it was viable and worked out. And one of the things that we realized at the time was the question, what is currency of collaborative work? So why should I contribute to such a paper? And the thing there is that some people say it's immediate use for me. I want to use the data immediately because I have a publication coming, like at Tina. Some say, okay, I have not something done at a meeting in the very moment, but I might do something in half a year, or I have a PhD student, master student line up who might come and do something. So it's good for us to have the data. And some people just do it because they like working in a group. So working in a group, feeling kind of part of a community, that is really something to do it. And it's also, like you see, we have 49 contributors across the globe that helped us with this project. And one of the other currencies, especially for the work we realized when talking to people, is having a recital resource you can actually add to your CV. So here's the bioRxiv print with the 17 co-authors we had from the first contribution. However, for example, for me doing this figure, it was declared in the beginning, oh, because it's a review paper by all the PIs, we don't want to have any kind of students on the paper, so we put you in the acknowledgements, which it's nice for honorable mentions, but you can't put it on the CV, so not really like a very hard currency to keep this speak. So after submitting it, we like to bioRxiv. We also properly try to publish it, and then they've got also the like next to it, also the paper that actually used the data, that was the initializer, came out on the here on the 11th of February of the whole analysis. And at the same time, we got also our own first citation out of it, because we were directly cited by the people. We have now a publication that we can use. And also it was even prior to the other paper published officially in Scientific Data with the same authors on it. And by now it has been cited already 15 times in the last 3 years. So it's a commonly used resource already. One thing that spin off after a few months of adding further data and thinking about it was the idea to stop chasing and start to implement something what I called the minimum information about ancient sequences. What do I mean? It is the biggest time bump which we actually realized there was that when you start extracting information from this paper, so you read through it, then they cite the lab protocol from another paper, which cites it from a third paper, and then they have some information in some supplementing material, which doesn't really have anything to do anymore with the part, or you have the part that some of the sample tables that have all the information are in a tabular format. However, in the PDF that was uploaded as supplementary material for the science, then you ask yourself, okay, what is going on here? That's really a thing. And then James had, for example, I found this one case. We have a study where James literally then said, okay, Diana tried, I also tried, but neither of us worked out this one out. Unfortunately, apparently multiple extraction occurred, but only 3 were sequenced, but only 2 libraries were uploaded to INA. So you have no clue. You might contact the authors. Some of them will respond to you, but it's a lot of wasted time. And so as a community, we said it would maybe sense to actually start standardizing the data entries. And we kind of already did that when defining what we want to add to the database in our case, where we said, okay, here we have, because many labs we have different standards of reporting metadata, and that vary very widely, we said, okay, you have to have certain things. So for example, site name as reported in publication, accents are allowed, missing name is unknown. Like I said before, latitude and longitude, decimal format, maximum 3 decimals. So if someone writes 10 decimals, we don't care, we round it to 3 decimals. If there's less precision, we actually shorten it, so fewer decimals if it's only roughly described. So we have like a codex of things. And by this, the main advantage of this for us was in the databases, if you search for a certain site location, you want to have sites from, like samples from a certain site, but there are 2 different spellings, one with accents, the other one's without. It gets very creative, so we more or less standardize the entries to actually allow us to make the database more searchable. And because of that, James and Antonio Fernandez from Copenhagen, who some of you might actually know, came up with this idea, why don't we actually propose to have a minimum information for ancient samples list that falls under the umbrella of this minimum information for sequences, this MIAX term. And there there's a list of things that have to be reported to actually when uploading things to INA. And the idea is to more encourage people to fill out the table, standardize that we can import the data machine readable in our databases and not waste someone's PhD students, postdocs, master students' time on digging through everything. And you already see down here, we have added for some of these names, these compliance fields where we already see that what will have to be added for the minor standard. At the moment, this is still developing. There's a work group that works on that, trying to do a publication. They reached out to the community there that is this genome standards community that curates the checklist and continues there. One thing we also realized is, and I call that release the beast, sequencing data information are added, which happened then in mid-2022, so more or less 2 years after we started, is that most for our data, I must say I'm very naive about proteomic data. I assume you have also some sort of public repository where you could, in theory, upload data. I think I heard from hearsay that they are not as well used as they are used for sequencing data yet, unfortunately, but I hope they are in the future. But mostly whenever you nowadays have to upload sequencing data, you have to provide it to one of the repositories. And the major ones are for the American folks is the NCBI SRA. We in Europe use the European Nuclear Archive or ENA. They actually, all of them are synced with each other. So if you upload the data, they already mirrored over to the other one. So they're double saved. And usually have this information there. And so what we realize is okay, I can go now to this data and download it, but for processing the data and knowing what the person have done, ENA doesn't record certain things. So for example, you might have heard that we in ancient DNA work, we are talking about like single-stranded library prep, double-stranded library prep, like UDG half, UDG full, UDG non-UDG treatment, and so on. This is usually not recorded. And so we said we will, for these different things, record this data additionally, and made, again, a general list of minimum information for library information that had information from the samples table already, like project name, publication year, sample name, archive, and so on, but then also very much information about how the sequencing data was generated and whether, like, that we know in the background to do it. And that's one thing that comes to the second part of my title. This, in the end, allows you to have very reproducible data analysis, because you can go there, you see it, you can automatize it, and so on. The almost final step was then this year in April was the automation and neural automation ancient metagenome DIR toolkit. What is the ancient metagenome toolkit? It originally derived as this kind of background checks that we had running in our GitHub automatically, like checking that latitude and longitude are numbers and things that I described. But then Maxime mainly puts the work in and developed it into a Swiss Army knife. So, for example, if you want to add samples, we declared we will only add samples to the database that are stored in a public repository. So otherwise, we're not interested. It can automatically pull this information from the public repository from using the autofill function for you, so you don't have to manually copy and paste all the things off the ENA database that I showed a moment ago. Additionally, it has a validate command, so that was the thing, double checking the the values if they make sense. It allows you to merge automatically data. It allows you to view, so this is the viewer part, and then it has a convert function. So developing new entries is, like I said, this idea that if you add new data and then it says, for example, here, my example from before, Scotland is not an accepted value for geolog name because it's not part of the list of all the countries that are here. You see here's a list of, we have a list of 280 or 70 something countries that are accepted according to this standard. And if it's not in there, and Scotland apparently isn't, it will flag it and will not allow you to add the data because we only want to have that. The person has to go back then and actually double check that and make sure that this works. The other cool thing is, and that's particularly interesting for people who are maybe not as tech savvy as, for example, James, Maxime, or I are, who like to really dig around and on the command line and filter data. But we have also provided a tool, this viewer command, where you see the whole database, and then you can say, I want to, for example, work on these 7 samples from Belize because I would be interested in that. And then I can, in the graphical interface, search for it. Here I searched for geolog name, Belize, got the data. I can validate the selection, and once I click the button, I can automatically download scripts to actually get the information on the libraries. I can get the scripts to download the data for me automatically, process the data automatically with pipelines. And we, especially James, Maxime, are working very much in the NF core. Sandra is a big contributor to many of them as well. So we have a lot of standardized pipelines that we provide automatic input in it. So you can more or less also, as someone who's not very tech savvy, directly process your data without having copy and paste errors because you might have missed it. And if you want to cite any of these studies, you can also download that part. Again, collecting rewards, we publish a preprint. this year in August that describes all of that at the F1000 research, which will then later, when the reviews are in, be converted in the proper publication. And the interesting part for us was we have, compared to the first publication, which was 2 years prior, a stark change of in the number of, or in the list of contributors. So only 8 of the people that contributed to the first study are still there, that are going on. And it's mainly the people around James, Maxime, and people at the MPI. But we also recruited 30 additional contributors from many different labs that were not involved at all in the original publication. So the number of labs really increased. And finally, just as a fruitful thought, what's the future of it? And that's also something that you would, I think, should consider when planning that. So here you see a graph. The nice thing about this, because GitHub keeps track of all when data is added, data is changed, you can see how much lines of text were removed of a file. You can and like added is green and removed is red. Mostly, what I want to pay attention to is the frequency. So we have here this rush in the very beginning where we add all the samples, then there's the publication, there's a bit of a downtime. And then from time to time, we have again this kind of spikes that quite often coincide if we have a hackathon or something like this, or just before publication. But there's extensive periods where there is no contributions at all, like here, then down here. And here, whenever, especially in the last few months, it has always, every 3 months we have a release of the new data, so official release of the data, kind of freeze the data set. And always the week before, we start actually working very actively on it. Like, let's push in because we have the release. But you also see that the contribution varies over the time. And one thing that James realized, and that I just shamelessly here screenshotted one of his Slack messages that he recently had, was that we actually realized that we drop off in the amount of contributors and submissions over the year. So we have, like, while we had a lot of momentum at the beginning and especially till mid last year, the last few months have been a bit stale. And the question is not really clear why. And we are hoping that with the feedback that we get from the contributors anonymously, why they drop out, we get a better understanding of it. As far as I know, so far the poll hasn't closed, so I can't reveal anything of that yet, even if I wanted to. But it's starting a project like this is one thing, but keeping going it is another thing. And I would have to say, if James would not dedicate so much time into it, we would have also not had so much success. So you really need someone who have thinks that this is very dear to their heart. To conclude the whole thing, first of all, I hope what I could show you today is that Git and like the version control tool Git and GitHub as a platform around it are the workhorses of collaborative research. So you should really make use of it if you try to do it, because it allows everyone around the world to access it, to collaborate with it. It doesn't matter if you sit in the same office, same building, same city or something. Everyone can, with a working internet connection, can help there. Secondly, you should invest a lot of time in teaching people and have also dedicated events where you try to train people to help community building, because then people feel more in like being part of the whole thing and going along there and helping out. And we really realize that these hackathons are really driving a lot of the move forward. You also should identify what are the rewards for the contribution to the fields, because a lot of people love to contribute to things. They're very happy to it, but there should be some kind of concept there that keeps them also is part of the community. We realize one thing is having like a citable source that we can put on our CVs and in form of a manuscript. So usually we try to make also bigger release, like every other year, a paper around release if we add a new functionality or something like this, and then also honor all the people that have been contributing in the meantime. And what I also can recommend is that once you have a resource like this, to actually make it also helpful, more spreads the visibility of it, that you should try to integrate it in either tools or projects, if like you have spin-offs of it that are built around this community resource. So we have here this as example, these pipelines that all are kind of integrated, can directly be used from the Ancient Metagenome Tier toolkit, you can directly access the data and processes with that, and then continue. And with this, I just want to acknowledge mostly James for doing all of that, being such a great leader of this project. Without him, that wouldn't be really the case. Tina for paying me for so many years doing all the cool stuff. The spamming community for all their contribution over the years, because like I said, I'm spokesperson now today in this talk, but not really it's not all my work. All the university affiliations that helped me over the years allow me to do the research, and funding bodies down here. And with that, I'm happy to take your questions if there are any. I hope at least there's some.
Thank you so much, Alex. That was a great talk. And I'm going to go ahead and stop the recording now so everyone just feels comfortable asking questions.